Hello everyone, welcome back again to the series of solved previous year's ICAR PG examination, Plant Science, where we will discuss the questions on genetics and plant breeding. This time we will solve the paper of 2018, so let's get started. Question number 1. Chloroplast contains disc-like membranous structure arranged in a stack, which is known as, grana. This is an individual granum which occurs in a stack. Question number 2. Breeding method to improve a specific character in a released variety is, it is called back-cross method. In this method, a desired character is taken from the donor parent, which is transferred to a recurrent parent or the recipient parent, and it is also known as the elite line. Question number 3. Which organelle is found with half-unit membrane? So the correct answer is option A, oleosome. These are natural oil droplets which are present as half-unit membrane. Cell lineage theory was proposed by Schleiden and Swan, which says that the cells arises from the pre-existing cells. Question number 5. Which of the following is a 5C sugar? So galactose, ribose, glucose and mannose, these all are sugars, whereas only the ribose has a 5-carbon sugar, also the deoxyribose. Rest, the glucose, galactose and mannose, they are 6-carbon sugars. Question number 6. Who found that a peculiar regularity exists in the ratios of nitrogenous bases or double-stranded DNA? So it was found by Erwin Chargaff in 1950, which was also known as Chargaff's rule, which determined that the purines are equals to pyrimidines, where A is equal to T and G is equal to C. Question number 7. Crossing over between the non-sister chromatids of homologous chromosomes take place during, it occurs during the Pachytene stage, where tetrad formation is seen. Question number 8. The megaspore mother cell is, it is a diploid which undergoes meiosis to develop 4 haploid megaspores. Question number 9. Seedless banana is an autotriploid having A and A genome, that is the 3 sets of chromosomes. Question number 10. Variation in diversity among plant population is influenced most by the taxonomic status. Question number 11. The cDNA libraries can be prepared by isolating. The correct answer here is mRNA. These cDNA libraries, they are constructed from mRNA that is isolated from a target fungal species at different development stages. You can see here isolation and collection of mRNA is done. Later it is amplified to build a library. The oxysomes occur in, they are present in the mitochondria, where they are responsible for the synthesis of ATP with the energy released during the electron transport system. Question number 13. First pigeon pea hybrid developed through GMS in India was, it is ICPH8. It is derived from the GMS line Prabhat and the cross of the male fertile line ICPL 161. First hybrid cotton developed in the world is, it is known as H4, which was developed by Chandrakant Patel, and it is an intraspecific hybrid. The Nick translation is directly involved in which of the following processes? It is involved in the conversion of DNA to DNA, where it is a technique for radioactively labeling of double-stranded DNA. Question number 16. Which of the following method is an ultimate source of creating genetic variation? So the correct answer is hybridization, where different species are crossed to develop a superior hybrid line. Back-cross method of selection is used for improving trait, which is the trait should be oligogenic. The one desired trait is transferred to the recurrent parent to improve the line. Question number 18. Most important organelle during germination, which is a characteristic of plant cell in the oil seeds is, so it is glyoxysome. These are specialized microbodies which later convert to peroxisomes in leaves when photosynthesis is initiated. Question number 19. Land races are, they are local varieties. The land races, they are cultivated and are genetically heterogeneous varieties which are involved in a certain area. Question number 20. Development of sporophyte without fertilization from the vegetative cells of gametophyte is called, it is known as apogamy. Question number 21. For which system of seed production the grow out test is a prerequisite for grant of certificate? The correct answer is option D, Csir system, that is chemically hybridizing agents. These induce artificial and non-genetic male sterility. A variety that has to be registered under Plant Variety Protection Act is referred to as, it is an extant variety. Question number 23. The cross-pollination of bajra is favored by, it is favored by protogyny. Protogyny is where the stigma, it matures before the anther, whereas the protandry, here the anther matures before the stigma. Question number 24. Prolonged rouging operation will not affect the genetic purity in case of, it will not affect the self-pollinated crop because the other seeds and the other chances of pollination are very rare in case of self-pollinated crops. Question number 25. The recurrent selection is mostly used in, it is used mostly in cross-pollinated crops. Question number 26. The following organelle also contains DNA. So out of these organelles given, only the chloroplast consists of DNA. A condition in which pollination and fertilization take place in an unopened flower is known as, it is known as cleistogamy. There is also a condition called chasmogamy, where after the pollination the flower opens and the fertilization, it occurs after the opening of the flower. Question number 28. A cell or an organism having one chromosome pair in addition to the normal somatic complement of the species is, so the correct answer is option number C, tetrasomic, that is 2n plus 2 condition. Monosomic means 2n minus 1. Nullisomic is 2n minus 2, whereas trisomic is 2n plus 1. A homozygous line developed by self-fertilization in a cross-pollinated crop is termed as, it is known as a pure line. Question number 30. Expression of both the alleles of a gene in F1 refers to, it refers to codominance. For example, the blood groups. Here both the alleles, they express themselves. In case of incomplete dominance, the dominant allele does not completely mask the effect of the recessive character. Thus, a mixture of characters is present. For example, the flower color of snapdragon. In RNA, the nitrogen bases are same as in DNA except, so the correct answer is instead of thymine, we have uracil. So A, U, G, C are the nitrogenous bases present in RNA. Name the enzyme which joins the broken DNA strands. The correct answer is option A, ligase. Question number 33. Cause for pseudodominance is. So out of these, the cause is the deletion. the deletion of dominant alleles occur where the condition of hemizygous occurs. It is a heterozygous condition, and a hemizygous is seen in case of pseudodominance where the deletion of dominant allele occurs. Question number 34. Match the following. Genetic male sterility, it is found in red gram. Cytoplasmic male sterility, it occurs in hybrid Napier. Genetic and cytoplasmic male sterility, it is found in sunflower. Parthenocarpy occurs in citrus, and non-viable seeds are present in onion. Question number 35. Match the following numerical aberrations. Nullisomic means 2n-2. Monosomic is the absence of one chromosome, that is 2n-1. Double monosomic is 2n-1-1. Trisomic is the presence of extra chromosome. Tetrasomic is the presence of a pair of chromosome, that is 2n+2. So that was it. Thank you.